good afternoon, everybody, and welcome to this next session, which is our Turbo Talk session. My name's Elaine Emmerson, and I'm going to be chairing this session. For those that don't know me, I'm a PI at the University of Edinburgh, and I work in the Centre for Regenerative Medicine. So today's session is going to be comprised of a talk from our sponsor first. So this is our sponsor, Fluicell. This is a recorded talk. So that will be first, then we'll move on to our keynote speaker, and then we'll have our turbo talks and then a collective question and answer session afterwards for all of those turbo talks. So without further ado, we're now going to share the recorded presentation from our sponsor, Fluicell.
Hello, my name is Tatiana Lobovkina. I'm the CSO of a biotech company, Fluicell. And today, I'm happy to present our work on generation of high-precision tissue models on solid and porous substrates. First, I would like to say a few words about our company. Fluicell is based in Sweden, and we offer products and services within life science field, drug discovery, and bioprinting. Our product portfolio includes bioprinter platform, Biopixel, which will be in the main focus of today's talk. Before introducing Biopixel, I would like to say a few words about bioprinting in general. So the core of this technique is similar to plastic 3D printing. However, the essential difference is that bioprinting operates by using a suspension of living cells instead of plastic filaments. So typically, the cells are housed in gel matrix or bioink, which is deposited using a layer-by-layer approach to build a tissue construct with the aim to imitate the behavior of native tissue. So to enable an optimal functionality of the printed tissue, a printing resolution allowing to control the cell-to-cell distances is necessary. So here at Fluicell, we address this challenge by utilizing microfluidic approach and generate high-precision tissues. This image shows the core of the Biopixel platform. This is a microfluidic print head which is inserted into a holder, and you can see a print tip right here. So using microfluidics for bioprinting allows to avoid usage of bioink, so the cells are housed in suitable culture media or buffer. And with this technique it is possible to generate high-precision tissue constructs in 2D and 3D with a printing capability ranging from single-cell arrays to multi-component tissues. It is also possible to generate tissue models on solid and porous substrates, which I'll be talking more about today. Our Biopixel technology conveys high-resolution cell patterning by circulating cells within a confined volume close to a substrate surface, maintaining them in native media throughout. The substrate can be either solid or porous, or it can be a confluent layer of cells. Through patterning of a substrate with the appropriate proteins, control over the 2D and 3D patterning can be established. This particular example shows formation of a 3-layer construct where we started with the preceded confluent layer of cells, on top of which we print cell attachment agent, which is required for efficient cell.
Hi everyone, sorry for the interruption, but it seems that we've got a technical issue with this presentation. So just bear with us a moment, and we're going to hopefully share this again so that hopefully people can see not just a blank or black screen. So bear with us a second. Hi everyone. Sorry, apologies again. We're again having some technical difficulties with sharing this presentation. So if you can bear with us a moment, we may just go straight to our keynote, but bear with us.
Hello, my name is Christine and I'm the chief editor of Nature Reviews Materials. And today I want to talk about scientific writing and publishing. So I want to give you some scientific writing tips and tricks that you can then hopefully use for your own research papers and reviews. And I also want to talk a bit about the editorial processes at the different Nature journals. So how we as editors assess manuscripts and how we take decisions. So what sort of papers do we look for? What sort of papers do we publish at Nature? So Nature itself publishes really important advances with wide implications, and the importance should be readily apparent to non-specialists, to researchers from different fields, and ideally to the general public. I guess we can all agree that, like, for example, the structure of the DNA is such an important advance, or the structure of the spike protein of the coronavirus. So these are the sort of papers that Nature is publishing. The Nature research journals do essentially the same thing, however, within their respective area. For example, material science for Nature Materials, or in nanotechnology for Nature Nanotechnology. So even if it's a biomaterials paper at Nature Materials, other material scientists should sort of understand that this is a really important advance in the general field of material science. So when we get manuscripts as editors, I want to talk a bit about the criteria that we have in our mind when we assess new submissions. So a key point is, of course, that it needs to be new. That means it should, of course, not have been published anywhere else, also not parts of it, but it also has to be new in terms of it needs to be a sufficient advance. And what do I mean with a significant, sufficient advance? It means that the paper should, for example, solve a long-standing problem in the field. Is this something researchers in this area have been trying to tackle for a long time? Is it a key pathway, a key synthesis, a key interaction that this work sort of understands or addresses? It could also be a change of our way of thinking about a question. For example, we've always thought this type of immune reaction occurs when we implant a certain material into the body, but this work now finds that there's actually a different protein involved or a different cascade involved. So it could be something like that, or it could also be, for example, a new tool, a new imaging tool, a new single molecule tool, something that not only researchers in your immediate area, but researchers from different disciplines could apply for their research. And it could also be a practical or clinical application. For example, if it's a material we've all been using for a long time, say lipid nanoparticles, and now you manage to use this material to deliver mRNA for a vaccine, for example, then this would be, of course, a significant advance because it would be an actual clinical application. So these are the sort of criteria we would look for. And then, of course, it needs to be interesting. So it needs to be interesting to not only people in your immediate area, but to a wide range of researchers working in similar or ideally even in other areas. It also has to be technically sound. although we of course rely quite heavily on peer review to help us with this. And there needs to be evidence for the conclusion, and with evidence I mean data. So every conclusion, every sort of key result, key finding needs to be backed up by robust data. So what makes such a great paper now? I guess, although there may be many answers to this question, I guess a fairly simple answer is great research. I strongly believe that it's very difficult to make crappy research into a great paper. So what do I mean with great research? It means there needs to be really strong evidence that lets you draw noteworthy conclusions. Therefore, a great paper really starts long before you actually write up a manuscript. It starts when you conceive the project, when you write the grant, when you write the fellowship, when you think about the research you want to do, the question you want to answer, the tool you want to develop. Is this a question that solves a long-standing problem in the field? Is this one of the big questions in the field? Is this a tool that would be super useful for many different researchers? And then, of course, it's important how you design your experiments. What animal model do you use? Which in vitro model do you use? Which cell lines do you use? And how do you analyze your data? What sort of statistics do you apply? So these are the aspects that really lay the foundation for a great paper. And then, of course, you need to write it up. You need to write it up as a manuscript. So, and when you write it up, it's important really to always tell the story, to write it very clearly and to know your audience. So it's very often good, I think, to take a step back before you actually write it up and think about what is the key message that I want to get across? What is my key result? And very importantly, who do I want this to read? So who are my readers? So if, for example, you have, say, an immunoengineering manuscript and you want to submit it to Nature Materials, it's very important that you explain the biology, the immunology in this context well enough for a material scientist to understand it. So you need to set the stage properly. Not only the editor should understand it and the peer reviewers, but your readers. Ultimately, you want many people to read your work, so you need to explain the context really well. So when you write up your manuscript, I think it's good to have sort of 4 key questions in mind that I'm sure every reader will want to know. First of all, why did you do your study? Why did you do what you did? And this needs to be clarified in the introduction. So first, there needs to be an explanation sort of the field. What do we know? What do we not know? Where's the gap? Where's the knowledge gaps? And where does your work come in? Why did you do it? What was your goal? What did you want to answer? And then, of course, what did you do? So your methods need to be very clearly and described and in detail described so that other people are able to reproduce your methods. So it needs to be clear exactly which cell lines, what animal model, which imaging parameters. So your methods need to be clear. And then, of course, what did you find? What are your key results? And then in the discussion, you want to state what your key results mean. So how does the study actually advance the field? What is the outlook? What could it be applied for? Where sort of are the next research step, or where do you go from here? So these are the 4 key questions to keep in mind when writing up your manuscript. And for the actual writing, it's important to really have a well-organized structure. Use paragraphs, make connections between sentences, assume that your reader knows nothing about it. What I mean is aim for a broad audience. Take a step back and think, did I explain, for example, the material synthesis well enough for somebody coming from outside the field to understand why this is such a huge step that I could make the material this way, for example. It's also important to really define all the uncommon terms, avoid acronyms as much as you can. Yes, you're going to get away with DNA and CRISPR, but keep in mind that acronyms may mean different things in different fields, and it just makes a manuscript very often very difficult to read. And in particular, avoid acronyms in titles and in the abstract, but also in the main paper. Explain the background, explain it clearly and simply. So if you can explain it in 1 sentence using simple words, it's probably a good idea to do this. We're not all native English speakers. I'm not, as you can probably tell. So it's good to have and sort of easy language to explain complex science. Be very clear. What I mean is avoid any ambiguities. It needs to be clear. Is this something that's common knowledge? Is this something that you speculate? So write it in a clear way and concise. There's so much literature published these days, so nobody has the time to read all of it. So if you can say it in one sentence, then say it in one sentence. And of course, figures are important. They are the backbone of your paper. So it is important that they are clear. It's more important that they are beautiful. Ideally, they are both, but it needs to be really clear. Having said all this, we would certainly never reject a paper based on poor language, but it needs to be clearly written. I've read lots of manuscripts at Nature Nano where I read it from the beginning to the end, and it still wasn't clear to me what the actual results were and what's actually the purpose of the study. Before you submit a manuscript, it's important to edit it. And this is an example of a review that I edited. It's a beautiful review by Adam Engler. We published it, and it's a wonderful article. But this is how every manuscript should look like before you submit it. So edit it yourself, get it edited by your colleagues or your PI if that's applicable. Go through it line by line, remove all ambiguities, remove all the empty statements that don't really contain any scientific information, make sure everything is explained well and clearly before you submit it. I also want to spend a slide on titles, because I really think that the title is the most important sentence in your paper, because this is how people will find your work. So whatever search engine they're using or whatever database they're using, if you look for an area, if you want to find answers or want to learn more about a specific field, you will use specific keywords to find it. And if these keywords that they're not in the title, people won't find it. So if you use a funny pun, which may be funny and good for, let's say, a news and user research highlight, but that means people may never find your work. So the keywords need to be in the title. It should be accurate. Of course, it's not fiction, it's science. It should be brief, clear again, declarative. So if you found a certain effect, don't just state there is an effect, state the nature of the effect. Something inhibits, something promotes. So it's important to be declarative. And of course, it should also be engaging. Don't alienate your audience, and be focused from the title. It should be really clear which field you're working in. And if you want to think again about your target audience, because it's I think it's such a key thing for publishing your work. In terms of target audience, the title should be broad. People outside of your discipline should really understand your title in terms of they know what field you're in and they know what your key result is. So aim for a really broad target audience for your title. Don't put any crazy transcription factor names in the title or something like that. People, or crazy acronyms that people may not know. Be really broad in your title. And then the abstract should also be fairly broad. The abstract is a mini version of your paper. However, it should really be clear, at least from the first few sentences, what the paper is about, and people should be able to understand it. And then the main text, of course, is for a more specialized audience. I want to quickly share with you the Nature abstract formula. So when I'm editing manuscripts and reviews, when we go through abstracts, this is usually a formula we always apply, and it works quite well to make abstracts more accessible, more readable. And so basically, you start off with 1 or 2 sentences giving a basic introduction to the field, very broadly. I'm working on biomaterials for bone engineering. And then you go into more detail. Specifically, I'm working on this biomaterial, or specifically, I'm working on this models. So in the first 2 sentences, keep it fairly broad and set the stage for the work. And then use 1 or 2 sentences to state the gap or the general problem that you are studying. So why did you do this? However, there is no material that can do X, Y, Z. However, it's been impossible to regenerate bone this way, blah, blah, something like that. And then it's important that your key result, your main result, has to be in the abstract, not any details of the methods. People can look it up in the methods, but your main result should be in the abstract. Here we show, here we developed. So put the main result in the abstract, and then use 1 or 2 sentences to talk about the bigger picture. So what does it mean? How does it advance knowledge in the field, and what could it be used for? What could it lead to? And once you have done all of this and you have your beautiful manuscript and your amazing research, you're going to submit it to hopefully one of our journals, and then it lands in our inbox. So I want to talk a bit now about what happens after you submitted it. So when you submit it to a specific journal, it will land in our journal inbox, and the editorial team meets a few times per week, usually going through all the submissions that came in, and then it's distributed, the manuscripts are distributed to the different editors, depending on the content of the manuscript and the editorial expertise. So at Nature Nanotechnology, for example, everything that had a cell or an animal or some biology aspect would come into my inbox. Sometimes it's 2 editors per paper if it's very interdisciplinary, but usually it's going to be 1 editor per paper. And the first thing then we're going to read is usually the cover letter. I know many people think it's just like an unnecessary requirement to submit a cover letter, but cover letter is actually a really good opportunity to address the editor directly, so you can use it to your advantage. So address the editor. Of course, if you know that the entire editorial team is female, it's probably a good idea not to write "Dear Sir" or "Dear Mr. Editor." If you know the editor, by all means use the name. Say what sort of format you want to have your manuscript considered as, and then you can pitch your work. It's not necessary to just copy paste the abstract, because we're going to read the abstract and the entire paper anyways, but you can pitch it. Provide a brief context. Say why is your finding is so important. What's your key finding? Why do you want the readership of this particular journal to read it? Why is it? Should it go into Nature Nanotechnology? What is so great about it? And then, of course, you can also include, suggest some referees, and you can exclude referees, and we will certainly honor the decisions. We would never send a paper to an excluded referee unless, of course, you've excluded pretty much everybody in the field who could review it, and also provide contact information. So the cover letter is a really good opportunity to address the editor. And if the cover letter sounds really exciting, we usually open the manuscript right away, because we get excited as well. And then we go through our editorial criteria that I mentioned before for every manuscript. So we read the paper in full, really from the beginning to the very end. We read the related literature. The better we know the field, the easier it is for us, of course. And then we check, is it original? Is it a new direction of research? Is it addressing one of these big problems? Is it one of the significant advances that we're looking for? Is it of broad interest? Will many people be interested in this? Or is it practically applicable and technically valid, of course? And based on this criteria, we'll then decide whether we're going to send it out for external peer review or not, or whether we reject it at this stage. And we select the reviewers based really on their technical expertise. They need to be able to understand and comment, of course, on the different technical aspects of the paper. Very often, if we know the field well, we know people that we can use. We want to have fair-minded, constructive referees, of course. We sometimes go with author suggestions. I usually look for my own referees first, and then if one of the referees I found is also one that's been suggested by the author, I may well go for this referee. We try, of course, to avoid any obvious conflicts of interest, which is sometimes difficult, especially now when we don't go to conferences and talk to people, so we don't know who works with whom, who just submitted a grant with whom. And we also want to have efficient referees, which is sometimes tricky. I mean, I'm sure many of you know that peer reviewing can be a bit of a bottleneck in terms of timing and to get a paper published. So we want to get timely referees, of course. And then we assess the referee reports. Most of the time we include 3, sometimes 4 referees. We never count the votes. If 2 say it's great and 1 says no, it's not good, it doesn't mean we necessarily go ahead with it. We really go through the reports and assess the argument, see if it's feasible, if it makes sense. And then we either invite revisions or would reject it at this stage. And sometimes we also suggest to transfer to a different journal. And we very often overrule or also reviewers' opinions if it's necessary. So I see this really as a sort of discussion between authors and referees. And as an editor, you're trying to sort of guide this discussion. And hopefully, at least from my experience, peer reviewing has made every manuscript better and the story more complete so far. Once you're invited to submit, do please address all the comments of the referees. You have a rebuttal letter, and you have, again, a cover letter where you can explain in case you think you cannot address one of the comments. Of course, always stay professional. Peer reviewers are your peers, so you probably know them or had a pint with them at a conference. And referees can make mistakes. So if you're unhappy with the decision and you think there are some, for example, factual errors in the peer review process, you can appeal at every stage of every decision stage. The papers will be seen again, appeals will be seen again by the same handling editor. And unfortunately, we can usually not give them the highest priority, so they often go on the bottom of our to-do list, so it may take some time for us to be able to get back to you, because sometimes it may be the better way of moving forward is to submit it to a different journal. In the last 2 minutes that I have, I quickly want to talk about Nature Reviews, of course, about my journal. We publish reviews, perspective, and comments, no primary research, and we do also a lot of editorial content. We edit every manuscript in detail, and we have a very strong focus on artwork. We have a dedicated in-house art editor who is redrawing all the figures in sort of the Nature style format, and we commission most of our reviews. However, you can propose reviews to us. So if you have a good idea, if you think, oh, now is the time that I write a review on this topic, you can please suggest it to us. It needs to sort of make us think, though, what a great idea. Yeah, there was so much primary research in the last 2 to 3 years. It really needs reviewing now. And when you write a review, it should really not just be a laundry list of everything that has been done over the last 15 years. No, it should be an authoritative assessment of a field. It should be accessible to non-specialist reader. If, for example, a PhD student comes in and wants to change field maybe a little bit for the postdoc, he or she should be able to read this review and get a good idea of where the field is, what are the key findings, and what are the open questions. The figures need to be good for a reviewer, scientifically complement the text, and it should be objective. It should not just cite your own research, of course. It should be a balanced discussion and forward-looking and inspirational, hopefully. So if you want to propose a review to us, you can write an outline, 2 to 3 pages with a tentative title, an abstract, what are sort of the main sections and subsections you want to review, what sort of display items would you like to have, what could be figures, boxes, tables, and it's important to be able to provide 20 to 30 key references of the last 2 years. If it's difficult to get 30 references from the last 2 years, maybe it's not the right time then to write a review on this. And with this, I'm running out of time. I just wanted to quickly flag the Nature Bioengineering community to you. If you haven't heard of it, look it up. It's like behind the papers, articles and pieces from editors, things that we find interesting, also scientific writing tricks and tips. So please do look it up. And with that, I want to thank you very much for your attention, and I'm happy to answer your questions. Thank you very much.
Great, welcome back everybody, and thank you for bearing with us through those initial technical difficulties. I just want to mention at this moment that we will share Fluicel's presentation at the end of this session, so after the Turbo Talks, so bear with us for that. And secondly, thank you so much to Christine for such an amazing presentation there. And I think that was really enlightening and hopefully a bit eye-opening for most of us in this conference. I'm just going to quickly introduce Christine because I didn't get a chance prior to her talk. So Christine received her MSc and PhD in nanobiotechnology from the University of Natural Resources and Life Sciences in Vienna, Austria, where she studied protein conformations and self-assembly using biophysical and theoretical approaches. Subsequently, she joined Molly Stevens' lab, who we heard from yesterday, where she worked to investigate extracellular matrix and cell material interactions. And then in latter years, worked at the Karolinska Institute in Sweden, conducting in vivo studies of antifibrotic materials. And as you've probably gathered, in 2017, she joined the Nature Reviews materials team as an associate editor. Subsequently, she moved to be a locum senior editor at Nature Biotechnology in 2019. And as of January in this year, she's now the chief editor of Nature Reviews materials. So it's my pleasure to be chairing this session and speaking to Christine today. So with that, I would like to open this session to questions. If anybody's got any questions, please do put them in the live question and answer. While we're waiting for a few questions, Christine, I've got a couple, if I could maybe start. So you touched upon the fact that the cover letter is actually much more important than maybe people consider. How does an editor make the decision between sending something out for review or not? Is the cover letter the most important thing, or is the manuscript, the abstract, for example, are there multiple things that are all important?
Thanks so much, Elaine. Thanks also for the introduction. The cover letter does not impact our decision whether we send the manuscript out for peer review or not. That's really the science. So the data and the science presented in the manuscript. And sorry, I hear some background noise here. Okay. Yes. So the science in the manuscript is the most important thing. As I mentioned, if it's a big question, if it's new, the cover letter is more, I think sometimes, for example, when I was at Nature Nano, it wasn't clear to me why a manuscript would come to us, why they would submit it to us, or maybe it wasn't as clear. If you're not that familiar with the field, it can be sometimes difficult to understand, is this really like such a huge thing or not? So in the cover letter, you can really make this pitch, and especially if you work in an area where you think, oh, maybe it's quite difficult, let's say, to get into a Nature journal because we're not publishing it as much, and you're like addressing the editor and say, like, look, this is a huge thing in our field, so please consider my manuscript. So, but the decision whether we send it out for peer review or not is really based on the science and on the data shown in the manuscript.
Okay, wonderful, thank you. So we have a question in the chat that says, thank you for your talk. After we resubmit the edited paper with the comments that we received, what can induce our paper to still be rejected? So are there occasions where all the comments have been addressed, but the paper can still be rejected?
I would say rarely. I think if really all the issues have been addressed, we would usually send it back to the referees because they are the ones sort of raised the issue in the first place. So they, in my opinion, should be the ones who also then comment on whether this has been sufficiently addressed. What happens quite often is that then there is another round of peer review sometimes. In KSC, the referees would say, yeah, you've addressed it, but for example, only partly. The reason we may not send it back to external peer review would be that we think, okay, you just haven't addressed the problem. For example, let's say the referees asked, really, you need to show this in a sort of in vivo or at least ex vivo setting, and you wouldn't do it. That could be a reason. However, we usually wouldn't reject it. We would probably reply to you and say, okay, I won't send it back this way unless you do a few further experiments. So just a straight rejection from a revised version is rare, I would say.
Okay, thank you very much. So I have another question. So you were talking about the fact that the paper will go to that specific journal that somebody submits to, and that there's not necessarily crossover between the different journals within the family. If the editor feels like the paper doesn't fit in their journal, but maybe does fit in one of the other family journals, does that get kind of communicated with the authors, and are there alternative ways to sort of sidestep to a more appropriate journal?
Yes, so we have a manuscript transfer service within the Springer Nature family, and when you submit a paper to one of our journals, you can you're automatically opted in unless you tick the box to say, I don't want you to discuss your work with any other editor, which I really would recommend you not to do, because that happens very often, actually, that we think is that is, oh, it's a good paper. It's just maybe not a Nature Nano paper, but maybe a great Nature Comms paper. And then we would consult with the editor at that journal, send this editor the referee, if it's already been out to peer review, the referee reports, or just share the manuscript and talk to the other editor about it and see what he or she thinks. Especially in our field, the editors, I think all of us at the Springer Nature family who handle like biomaterials, bioengineering, tissue engineering content, we are like 6-7 people, so we know each other very well. So I know quite well what Robert, for example, from Nature Comms, or what Pep and Nature Biomedical Engineering, what like his gap is. So if we would suggest it, it's very likely that they think, okay, I'll consider it. So yes, that's a good option, I think.
Okay, super, thank you. If anybody has any questions, please do put them in the live Q&A. I guess I would also like to ask a question that's maybe a little less about necessarily your role as an editor, but in terms of if this was a career path that somebody was thinking of taking, is there any advice that you can give anybody in terms of taking this sort of path after either a PhD or a postdoc?
Yeah, I mean, first of all, it's a fantastic career path in case somebody in the audience thinks of, oh, I love science, but I'm not sure if academia is for me. Because as an editor, you're still sort of part of the scientific community. The main thing you do is you read and think about science and research. You don't do it yourself anymore, of course, but you're very much in this sort of scientific academic sort of environment. And if this is something you would like to consider, I think it's good to get as much involved into the publishing process as you can. Writing papers, maybe getting involved in peer review. Maybe you can ask your PI if you can, if he or she can mentor you with the peer review process. If you can get involved in drafting rebuttal letters, like all the stages sort of publishing. Also, of course, writing. Any science communication, even if you're very active on Twitter, that's a good thing to get involved in science communication, because as an editor, you will write editorials and research highlights and blogs, and we do quite a bit of writing. So I think that's the things you can do. And usually for roles at the Nature journals, we would mostly ask for a postdoc. However, sometimes if you have done a PhD and you work in a certain area where we're really looking for an editor who can cover this area, by all means, please do apply if there's something you think could be a good fit for you. But I guess the main tip is really to get your hands on as much reading and writing, science writing as you can, and also to stay fairly broad, because as an editor, you will handle areas that are quite outside of your comfort zone sometimes. So the more you read, the more you know, of course. I guess that's the main tips I can think of now.
Great, thank you so much. And just one final question that's coming in from our Q&A. What are your thoughts on post-publication peer review platforms such as PubPeer? How do you see the field of scientific publishing evolving in the future?
Yeah, and that is a very difficult question to answer, but I'll do my best. I think it's good to have papers out there and people can comment on it. To me, it is a bit different coming in now from the other side. It is a bit different, the review or process than a peer review process, because and I'm sure many of you act as peer reviewers as well, quite often for journals. Because you get the manuscript, you really read it in detail, you really think about it. Usually the referee reports I get are really constructive, they're really good. So you can tell that the researcher has really tried to be constructive and try to make the paper better. And sometimes comments that are on PubPeer or that are on online platforms are quite broad, or sometimes where it's like, I don't even know 100% who this is, what this means, because we pick referees really based on their expertise, so we can be confident that they really know what they're talking about. In terms of the future, scientific publishing is changing a lot at the moment. I mean, there is open access, there is guided open access, there is preprints, and I think that's good, because I think publishing should change. Science has changed, and science is constantly changing, and has changed now like crazy during this pandemic. I mean, we have done things so fast like never before. People have collaborated, industry, academia has collaborated. We published papers within 3 weeks because we know it's important coronavirus biology. So things are changing, and I think that's good. Where it really goes, who knows? I think editorial roles and some form of reviewing will be part of it, because I still think it's important to have some sort of filter or somebody like me who sits at home, and my main concern is to make sure that the paper we publish is correct and that it's good and that people have access to it and there's going to be a press release and people will read it. However, in which form, yeah, if I could look into the future, that'd be great, but I can't. But I think it will change in a good way.
Great, thank you so much. Thank you for everybody that's posted questions, and I'd just like to thank you again, Christine, for that really insightful talk, and thank you for joining us today and giving us your insights.
Thank you so much, Elaine. Thank you, everybody.
Thank you. Great, so we're now going to move seamlessly on to the Turbo talks. So we're just going to go through the talks with no questions after, because these are just 4-minute talks. And then we'll have a collective Q&A at the end. So if you're thinking of questions during these talks, please do start posting them in the chat straight away, and I'll keep an eye on those. We have had a couple of technical issues, so there might be some pre-recorded talks, but I believe that our first speaker, who is Jonathan Hinchliffe from the University of Sheffield, I think is able to present live. So over to you, Jonathan.
Yeah, thank you very much. My presentation is on the development of a next generation natural polymer-based bioartificial pancreas for the treatment of type 1 diabetes. And my supervisors are Professor Ipsita Roy and Dr. Nicola Green. So in general, the pancreas is obviously an incredibly important organ. There are two main functions it has. It has the more digestive component, or the exocrine component, which comes from the acina cells. And then through the beta, alpha, and delta cells, you have this endocrine component, which helps in blood glucose homeostasis. So this is all very well and good. Obviously, during diabetes, this is when the machinery of your body goes wrong. So usually upon the first exposure of an antigen, your macrophages would become antigen-presenting cells, which would then present these antigens to cytotoxic T cells, and therefore activate T cells in order to induce a cytotoxic response against the foreign material, and consequently release memory T cells, which would be able to form and mount the same response on a second exposure to that same antigen. However, during diabetes, there are certain molecules that are expressed by beta cells in the islets of Langerhans, such as proinsulin and glutamate decarboxylase, which the immune system is sensitized to, and therefore mounts an immune response, and therefore mounts a cytotoxic response against your beta cells, causing beta cell tissue loss, and therefore an inability to produce insulin. So, how do we solve these problems? Well, the first idea that people have come up with is immuno isolation, which is a process by which you take a therapeutic material, in our case, encapsulated beta cells, and what we do is we encapsulate them in a protective membrane, which is semi-permeable, which allows insulin, glucose, oxygen, nutrients in and out, whilst stopping antibodies such as immunoglobulins and also the cells of the immune system from entering and killing the beta cells. In our lab, we couple this with a class of polymers known as polyhydroxy alkaloids, which are a class of non-immunogenic and relatively biocompatible polymers. And we found that they have a lot of good degradation characteristics and incredible tunable mechanical properties, depending on the tissue that you want to use. So in the lab, using a fermentation process and also using a specific species of bacteria, you can use Bacillus species, Cupriovidus species, and also in our lab, we're using Pseudomonas species in order to produce these PHAs, from which we take from a fermentation. We then centrifuge out the cells, freeze dry it, and then by purifying it, we end up with pure bacteria-derived polymer. So our idea is to take sodium alginate, which is another well-known non-immunogenic material and one that has been used in immunoisolation for decades, and combine it with BRIN-BD11, which is a pancreatic beta cell line which produces insulin. And then what we can do is we can combine this with our polyhydroxy alkanoates, in this case PHOHD, which acts as a kind of mechanical cage around the construct that we're making, which allows the alginate to function as it should without mechanical degradation, because sodium alginate hydrogels are notorious for not being that mechanically strong. And we're able to do this using multi-material fused deposition modeling, 3D printing. So here's just some little images that I've got of the 3D printer at work. You can see the HOHD being printed here. And then you can see in the next video, you can see individual blobs of alginate being printed. Now, bearing in mind, this isn't the final design of a construct, but certainly this gives us a good idea of eventually what our construct may eventually look like. And you end up with a structure like this, with single blobs of alginate that are protected by an elastomeric polymer. So what we found is that there's no general degradation of the alginate over 7 days, and we've also found that the structure retains its mechanical stability, though of course we'll need further mechanical testing to confirm its functionality, and also when we eventually do bioprinting with this. in terms of looking at a little deeper view, we did some SEM, and we found that the structure remains very similar to the CAD design models that we wanted, with a very good construct displaying alginate within the PHA grid that you can kind of see in some of the wells. Though, of course, we'll need to do further EDX characterization and also environmental cryo-SEM in order to really see the alginate in its geometry properly in situ within the grid that we've made here. So in order to further improve this, obviously once we've bioprinted it, in order to further improve the viability of the cells within the construct, there are various things you can do. For example, we could increase the porosity by doing a technique called sugar or salt leaching, which would allow us to make our cage a lot more flexible and also similar to pancreatic tissue. Other things we could do is we could functionalize either the HOHD or the alginate by impregnating it with oxygen-generating molecules, which would allow us to reduce the hypoxia that seems to be an issue in microencapsulated structures. Obviously, that just treats the problem of hypoxia, not its source. So prompt revascularization would also need to be encouraged. So, for example, we could use factors like VEGF-A and BFGF, which encourage the growth of blood vessels to restore this islet oxygen supply promptly. And then finally, obviously, because diabetes is an immune disease, we could use localized immune suppression by taking factors such as TGF-beta and also anti-inflammatory interleukins such as IL-2 and 10. And what we could do with those is we could leach those out of the construct, and not only would this increase the survival of T regulator cells, but would also upregulate the production of proteins such as perforin, which actively target T effector cells and therefore regulate their function. So that's a nice little picture there. So in conclusion, whilst type 1 diabetes does have different causative elements, obviously its effects require constant insulin replacement. Immuno isolation can help with this problem by replacing that damaged islet tissue with less risk of that immune rejection, as it is isolated from the body. The PHAs and alginates that we've been using remain excellent candidates for a relatively biocompatible but also non-immunogenic solution to make a semi-artificial pancreas, though of course ourselves and other people working on this need to determine the kind of construct biocompatibility and also the effect of future functionalization on the viability of the cells contained within. I'd like to thank my supervisors and also Dr. David A. Gregory and the Roy Lab for providing me with some excellent photos. And thank you very much.
Great, thank you so much. That was a lovely start to our Turbo Talks, so thank you very much. Right, so the next talk is from Roxana Sava from the University of Manchester. Unfortunately, we've had some technical difficulties here, so this is going to be the recorded version.
First of all, to thank to the organizers for providing this opportunity to present the work we are doing in the brain inflammation group on developing a regenerative therapy for brain repair and functional recovery after ischemic stroke. Most of us have an elderly family member or at least heard of someone suffering a stroke. And this is because it's a very common disease, which can be fatal or can leave the surviving patients with serious disabilities, thus representing a huge economic burden for the health systems. But what exactly happens during an ischemic stroke? First, a brain blood vessel is blocked, and this causes a reduction or interruption of blood flow. This initiates a massive inflammatory response, brain tissue death, and in the end, the dead tissue is replaced by a cavity. As a result of the brain tissue loss, patients present a functional impairment with major consequences on their quality of life. The only approved treatment, recombinant tissue plasminogen activator, is only available to a limited number of patients, being effective only the first few hours after the stroke onset. There are no treatments targeting the later stages, which encourages the exploration of potential regenerative therapies. Following stroke injury, the tissue repair is limited by several factors, such as the lack of structural support, inflamed microglial cells, which are the brain resident immune cells, and insufficient endogenous repair processes for new brain tissue growth, like angiogenesis and neurogenesis. These limitations could be overcome by using hydrogels for supporting new tissue formation and for delivering pro-regenerative therapeutic agents. We envisioned a potential therapy based on synthetic self-assembling peptide hydrogels. These are fully defined materials made of non-toxic amino acids and can form non-covalent interactions with proteins, property which can be exploited for the use as delivery systems. Their injectability and in situ gelation also ensures a minimally invasive administration. So what we propose is a combination therapy based on two therapeutic proteins, THG6, a microglial modulator, VEGF, an androgenic and neurotrophic factor, and the commercial self-assembling peptide hydrogel, which contains ArchD and IgVAV, cell adhesion motifs, and has the purpose of offering structural support and act as a delivery system. To test this potential therapy in vivo, we are working with a mouse model of ischemic stroke, and hydrogel loaded with THG6 and VEGF is injected locally into the stroke lesion in the subacute time window, when most of the inflammation has been resolved and the regenerative processes start to happen. Then we are looking at both tissue level and functional repair. This hydrogel has shear thinning properties and releases VEGF in a sustained manner in vitro for at least 5 weeks. Moreover, 48 hours after intracerebral injection in mice, the hydrogel induces significantly less microglial activation, less astrocytic scarring, and less phagocytizing cells compared to control, suggesting it's biocompatible with the brain tissue. And this data encouraged us to use RGD-12 peptidogel alpha-2 further as a VEGF delivery system for stroke therapy. Apart from VEGF, we are using THG6, which modulates the microglial phenotype from pro-inflammatory to pro-regenerative and has shown great potential in other brain injury animal models. After stroke, we saw that THG6 administration into the lesion subacutely has shown a trend toward improved animal well-being long-term, as reflected by the burrowing test and the animal's weight. At tissue level, we saw a trend in decreased microglial inflammatory activation and a significant increase in new endothelial cells in the stroke core, suggesting an androgenic effect. Finally, our ongoing work is testing the reparative effect of the proposed combination therapy in the same mouse model of ischemic stroke. At 28 days post-stroke, we will look at tissue level repair, more specifically markers of brain inflammation, glial scarring, angiogenesis and neurogenesis, and functional recovery, so differences in cerebral blood flow and performance in behavioral tests. Finally, I would just like to thank everyone involved in this project, my supervisors, colleagues from the Brain Inflammation Group, to our collaborators, to my funders, and to you for listening.
Great, thank you very much for that talk from Roxana. We're now going to move on to the next talk, which is Alessandra Grillo from the University College London, and this is also going to be a recording.
Hello, everyone. My name is Alessandra Grillo, and I will talk about my project, which is bioengineering a 3D tissue model of the rectus sheath to study herniation. Hernias are protrusions of the abdominal organs outside of the abdominal cavity, and almost 20 million hernia repairs are performed every year around the world, with 100,000 just in UK, causing chronic pain and inflammation in most patients. The layer mostly affected during herniation is the posterior rectus sheath, a connective tissue formed by the aponeurosis of the anterolateral abdominal muscles. This tissue presents a highly anisotropic structure, with bundles of collagen fibers running along the transverse plane, as shown in the anatomical magnification. So, the aim of this study is to develop an in vitro 3D model to mimic the rectus sheath and study herniation. This model is made with type 1 rat tail collagen, which is neutralized and set at 37 degrees. The excess fluid is then removed using the well-known draft technique, combined with mechanical shear flow applied on the longitudinal direction to align collagen fibers and obtain an acellular flat sheet of aligned collagen. Then, to test that cells would actually align accordingly to the pattern presented on the aligned gels, and also to verify that collagen construct was actually aligned in its fibers, We seeded HDFs on the gels, cultured them for 4 days, and imaged with fluorescence microscopy to quantify cellular alignment, and imaged with second harmonic generation microscopy to quantify fiber alignment. Results from the fluorescence microscopy show that human dermal fibroblasts sense the aligned structure of the gels and align accordingly, comparing to the unaligned control gels where cells are randomly distributed and aligned. Quantification of these data also confirmed that aligned gels have a significantly lower mean angle of deviation from the horizontal comparing to the unaligned ones, meaning that they are mostly oriented along the direction of alignment, indicated by the yellow arrow. To confirm the alignment of collagen fibers on the samples, second harmonic generation was performed, and again, there is a clear difference between the aligned and unaligned gels. Histogram representing the frequency of fiber angles show a flat curve for the unaligned gels, which represents a random pattern of alignment, and a peak for the aligned gels very close to zero, meaning that there is a higher frequency of horizontal fibers in the horizontal axis, which is the direction of alignment, according to the frame. So the take-home message is that we developed a tissue model which mimics the structure of the rectus sheath. Next step will involve to seed primary healthy and hernia fibroblasts from the rectus sheath to study the biology of herniation and their interaction with the model. Finally, we created a model which can be adapted and utilized to study different aligned tissues like tendon, ligament, cornea, and this just by tuning simple parameters like dimensions, density, and thickness. Thank you for your attention.
Great, thank you very much for that, Alessandra. So our next talk is Busra Baskapan from the University of Edinburgh, and she's going to be presenting live. So over to you.
Hello everyone, hope you can see my presentation and hear my voice very well. I'm a PhD student working on kidney tissue engineering based on Kalman's group in Bioengineering Institute at the University of Edinburgh. And I'm presenting one of my studies today. So first, a little bit background. As you may know, 10% of the world population suffer from different level of kidney diseases. And in UK level, around 3 million people have chronic kidney disease. Only treatment options are dialysis and transplantation, which called renal replacement therapy together. And as you can see in this figure, the incidence rate of this therapy has risen over 10 years. This means every day 20 new people in the UK has developed kidney failure. And it also brings a financial burden on society, as you can see in this figure too from NHS, which shows on the whole around £1.4 billion is spent on chronic kidney disease therapies. So to find an alternative solution or improve the options we have, we are looking at hybrid approaches in kidney tissue engineering, especially in terms of cell material interaction. And in this particular study, we produced hybrid scaffolds from polycaprolactone and laminin via emulsion electrospinning, where a surfactant is used to form emulsions in the solution to protect protein from solvent damage. So we obtained scaffolds with increasing concentration of laminin, as you can see in these SEM images, which show quite homogeneous structure morphology with almost same fiber diameter and quite similar mechanical properties between groups. Then we also look at cell response to the scaffolds. We cultured RC124 kidney cells over 2 weeks. And briefly, biochemical analysis show that laminin addition has an impact on cells, especially viability, with the highest concentration of laminin within 24 hours. This was also supported by the DAPIan followed in staining of the cells on scaffolds, which reveals that cells favor scaffolds with laminin, especially with an increase in concentration and over time. We also found that gene expression is affected by these hybrid scaffolds with a key marker MPEP. And this gene is associated with tumor formation, where a downregulation was noted, which points out a better adaptation and healthier function of cells over time. So on the whole, briefly, we were able to show that the hybrid scaffolds affect the kidney cell response and the importance of these hybrid approaches in kidney tissue engineering. We will be looking at deeper into this system in the future. Lastly, I would like to thank all of my group members, present and past, for their help. And if you're interested in, please come to my poster number 28. Thank you.
Great, thank you very much for that, Busra. So next we have Lydia Beacon from the University of Nottingham, who I believe is also joining us live. Sorry, apologies, Lydia couldn't attend today. So we're moving straight on to Veronica Hidalgo Alvarez, and this is a recording. Oh, sorry, this is Lydia's recording. Apologies.
Hi everyone, I'm Lydia, and today my poster flash is on assessing the therapeutic potential of corneal mesenchymal stem cells for ocular surface disorders. So currently, if you have a chemical or physical trauma to the cornea, it can lead to a really aggressive inflammation. And this is where my project comes in, because we've been looking at regenerative medicine therapies to combat that acute phase of inflammation following corneal trauma. And the main idea of this is that it would bring down the waiting times for corneal transplant. So the idea is that we take mesenchymal stem cells, apply them topically to the cornea, and these cells would ultimately release trophic factors to lead to a dynamic response and wound healing. And the MSCs that we've been using are corneal mesenchymal stem cells. So these are derived from corneoscleral rims, which are a waste product of transplant surgery. So we take them from this dark region here, which is called the limbus. In terms of the main aims and methods of my project, the overarching aim is really to characterize the cells because they're quite a novel cell type. So we started by doing some CMSC phenotypic profiling to assess the donor-donor variation. We then went on to compare these results to MSCs from other sources, for example, bone marrow, which is the current gold standard. And finally, we wanted to assess the cells in the CMSCs, including their phenotype and behavior in an in vitro inflammatory environment. And that's because the first two aims were done with the cells on tissue culture plastic in quite a happy environment. We wanted to see how they would respond to that toxic ocular surface, toxic injured ocular surface. So first of all, with the phenotypic profiling, we used a BD lyoplate. So it looked at 242 different cell surface markers. And we found quite a few of the markers did display high heterogeneity across the 5 donors, but also there was high homogeneity across some of the markers as well. And we could deduce from this that these markers should be present on all CMSCs or not present. Our second aim was to compare CMSCs to MSCs from other sources. And I've just got one example here of a paper by Amati al. So you can see here on the left, and he compared MSCs from umbilical cord and bone marrow. And what's nice is if we look at our graph on the right, that you can see that they're displaying a really similar phenotypic profile. Okay, and then finally we went on to look at the MSCs in that in vitro inflammatory environment. So we added an inflammatory cocktail, and we found that although there was a drop-off in the viability of the cells, you can see here fewer cells in the live dead, this subset of cells did release secrete higher levels of anti-inflammatory, pro-inflammatory, anti-angiogenic and wound healing markers. So you can see that they are behaving differently in this environment. And then finally, we wanted to look at the phenotype, almost do a full circle phenotype of of the cells when they're in this inflammatory environment. And we looked at treated and recovered cells. And we found that some markers were maintained throughout all three groups, some changed during treatment and were maintained through to recovery, and some only changed for treatment, but then returned to baseline following recovery. So this starts to give us an insight into what's happening to the cells, and also any benefits or disadvantages to priming the cells prior to their application. So in summary, we have looked at phenotyping the CMSCs. We've also performed a comparative analysis to MSCs from other sources, including bone marrow. And finally, we have assessed the behavior and the phenotype of CMSCs in that inflammatory environment. Thank you very much. If you would like to know more, please come and have a look at my poster.
Great, thank you very much to Lydia, and apologies, that was my mistake. Lydia was unable to attend, but we did have her recording. But we're now moving on to our next speaker, Veronica Hidalgo Alvarez from Heriot-Watt University, and she'll be presenting live. So over to you.
Hello, everyone. I'm a postdoc in Pharmacos group at Heriot University, and today I'm going to talk about the development of polymeric capsules for single administration vaccine enhancement. Vaccination involves administration of a first vaccine dose, followed by a booster dose after weeks or months in order to enhance immune response. The need for a second dose can pose a challenge due to the extra cost involved or any logistical issues, or in the case of animal vaccination, the difficulty to recapture a wild animal in order to give the second dose. Therefore, in a project, we are developing polymeric implantable capsules for the delivery of this second dose. And this is achieved by encapsulating an osmogen together with a booster dose in order for the release to be driven by osmosis. So after the implantation, the osmogen causes an increase in the water uptake, which leads to a rise in the hydrostatic pressure, and this makes the capsule burst at a specific time, releasing all the payload at once. The capsules are made by dip coating, which is a process in which a metallic rod is repeatedly dipped into a polymeric solution. The resulting coating is then detached, post-cured, and filled with the second vaccine dose and the osmogen. And the capsules are then formed by sealing the open end of the tube. In this work that has been published recently, cross-linked and non-cross-linked PCL were tested as materials for the synthesis of the capsules, and a dye was encapsulated in order to evaluate the release kinetics. As you can see on the left, the mass of the capsules increased substantially during the first 20 days due to the water uptake. This was followed by the burst, in which part of the payload was released. And after this, there was a period of sustained release. At burst, the capsules made of cross-linked PCL released around 9 times more dye than the capsules made of non-cross-linked PCL. However, the maximum amount of dye released at this point was only about 10% of the total payload. And as you can see on the right, even after the period of sustained release, only around 40% of the total payload was delivered by the capsules. Therefore, improvements were needed on the formulation, and to achieve a higher release at burst, a fully amorphous copolymer was developed, which was composed of polycaprolactone and polylactic acid. So as you can see in the graph, with this the capsules made of this material released around 60% of the payload at burst, which was a significant improvement compared with previous formulations. And this was due to the elastic recovery of the material. So going back to the PCL formulations, the toxicity was tested in vitro. And as you can see in the graph, the material was not toxic, and the metabolic activity at day 11 of the cells seeded on PCL was higher than that of the control, which is depicted here with the dashed line. During the first 4 days, as you can see, the metabolic activity was lower than the control, but this was due to the delay in cell addition to the PCL materials, as you can see on the images on the right. So these experiments demonstrate the reliability of osmosis as a mechanism for the delay in the release of the payload from the capsules. Further formulation development was required in order to increase the release at burst, and the results showed the efficacy of the use of an amorphous copolymer to achieve this. The studies performed in vitro confirmed the lack of toxic responses by the polymer, And the future work will focus on the automation of production, the quality control, and the method of administration to patients. And with this, I'd like to thank all the members of the project and of the group, funders, and thank you for the attention.
Great, thank you very much, Veronica. So now we're moving on to our final speaker in this session, Krit Ratanawa Wansakul. Apologies for that terrible pronunciation, from the University of Sheffield, and over to you.
Thanks for the introduction, Elaine. Good afternoon, everyone. I hope you can hear me clearly. I'm Krit. I'm a PhD student at the University of Sheffield, and my topic today is on the effect of geranyl or GGOH on solidinate-induced toxicity on oral fibroblasts and keratinocytes. So I would like to give you a brief introduction about this work first. So the solidinate toxicity on oral fibroblasts and keratinocytes is related with the condition we call the medication-related osteonecrosis of the jaw, or MONJ. It is an adverse effect of anti-resorptive or anti-angiogenic drugs, mostly found in patients who receive bisphosphonates for osteoporosis and bone-related cancer treatment. The main clinical presentation of MONJ is an exposure of necrotic bone with incomplete soft tissue healing in the oral cavity. The majority of MONJ cases are patients who using nitrogen-containing bisphosphonate, such as solidinate or pamidronate. This bisphosphonate primarily inhibit the mevalonate pathway, causing the reduction protein synthesis, which are essential for osteoclast activities. Besides the detrimental effects on osteoclast, the toxicity of bisphosphonate also impairs oral mucosal cells. So up to now, the management of MONJ is still a challenging problem for clinicians, and the standard treatment procedure has not been determined. Research is currently focused on developing alternative therapies to treat MONJ patients effectively. And one of the possible candidates is the geranyl or GGOH, which is a derivative granulate molecules in the mevalonate pathway. This molecule has been able to counteract solidinate toxicity in osteoclasts. So we hypothesized that GGOH could also protect oral soft tissues from bisphosphonate toxicity as well. So this study aimed to investigate in vitro effect of GGOH on solidinate-induced toxicity in oral mucosal cells. So two types of cells are used in this study, which are the primary oral fibroblasts and keratinocytes isolated from buccal mucosa from volunteers. And cells were incubated with GGOH concentrations from 0.5 to 10 micromolar alone, or GGOH in combination with 10 micromolar of solidinate for 72 hours. Then the viability of cells was then analyzed using an MTT assay. And we found that GGOH alone, even at 10 micromolar, had no significant toxic effect on cell viability after 72 hours of treatment. And solidinate markedly lowered the metabolic activities of keratinocytes. For the rescue effect, the GGOH slight recovery in the viability inhibition of solidinate in dose-dependent manner. However, there was no statistical significance was observed. So in conclusion, GGOH produced no cytotoxicity towards oral fibroblasts and keratinocytes, but there was no apparent effect of GGOH on the reversing solidinate toxicity. In future, we plan to perform the experiments using pamidronate or higher GGOH doses to confirm the efficacy of GGOH and its potential to be used for MONJ management. I would like to acknowledge my sponsor, the Faculty of Dentistry, Mahidol University in Thailand, or my supervisor, Dr. Vanessa Herden, Frederick Clayson, Robert Bolt, and Simon Atkins for good advice and support for the project, all Herden's lab member, and Vanessa Singleton for laboratory support. And last but not least, Dr. Helen Colley for the support in obtaining primary cells. So if you have any question or would like to discuss about this work, I'm very happy to answer and also chat during a post session after this. Thank you for your listening.
Great, thank you very much, Crit. And I'd just like to take this opportunity to say thank you to all the presenters in that session. That was a really fantastic, quick overview of all of their research. And I'd really like to encourage everybody to go and visit their posters in the next session. We do have time for a quick Q&A, so I think we're going to bring all of the presenters back now. Fantastic. So please do put any questions that you've got in the Q&A, in the live Q&A box. I can kickstart with a question for Roxana. So I wondered if the injection of the hydrogel itself into the injured brain causes any further injury or any further inflammation?
Yeah, so that's a good point, and kind of everyone in the field is concerned about that. So regarding the volume of hydrogel, we definitely know that the volume we inject, which is 6 microliters in a mouse brain, does not cause intracranial pressure. Regarding the injury, we are using very fine needles, so there is a very small amount of injury, basically, but it's not very serious, so it's acceptable.
Great, thank you very much. We have a question in the Q&A that I think is possibly for Jonathan. Okay. The question is, how do the scaffolds prevent diffusion of immunogenic molecules? Does it actually filter at all?
So when it comes to making like a material that is immunoisolatory, there's a very small window of opportunity. So in terms of pore size, so you want something that is going to be with pores small enough so that the cells of the immune system can't get in. So this is usually between 15 and 18 microns. And then you get a little bit smaller, and I believe the minimum or the maximum size you can make your pores is roughly, I think it's about 7 nanometers, because that's about the size of an immunoglobulin molecule. Luckily for us, insulin is actually obviously a smaller molecule than that, so it's not really there are I know of chemical ways of filtering it, but luckily for us, alginate's porosity is naturally able to let insulin in and out whilst blocking most, if not all, immunoglobulin. So it's mostly a size-based filter more than anything.
Great, thank you very much. So we've got a question in the chat for Busra. How do you achieve multi-protein approach for your scaffold?
Thank you for the question. And I'm sorry for my laptop's fans they bursting because of camera. And the question is a very good question. Actually, we didn't try different proteins or multi-protein, but I think there is a good way to do this using the decellularized tissue, because basically it contains a lot of different proteins and basically everything except cells. So the next step for me will be to try this and see the results in this emulsion system. So it will be a multi-protein approach in the future.
Great, thank you very much.
Thank you.
Okay, and a final question for Crit. So I work on saliva, so that's something that's really interesting to me. Do you know if the saliva or any of the enzymes that are found in saliva have any role in osteonecrosis, either positive or negative?
It's an interesting one, but I'm not quite sure on that. On the enzyme from the saliva that may be the part of the development of the osteonecrosis or not, but due to the very complex of the pathophysiology of the MONJ, it may be developed from like the infection or the inflammation, may be like something on the drugs that we see may be present in maybe in bone, or like has a direct toxic on the soft tissue, or like maybe something in the saliva that may also contribute in the development of the osteonecrosis as well.
Great, thank you very much. Well, I'll just take this opportunity to thank you all for your brilliant presentations. I'll encourage the audience to please go and speak to people at their posters and go and see the presentations. And finally, we're just going to move and try and present the sponsor recording again. So this, just to remind everybody, is from Fluicell. And again, please go and see them at their booth as well.
Hello, my name is Tatiana Lobovkina. I'm the CSO of a biotech company, Fluicell, and today I'm happy to present our work on generation of high-precision tissue models on solid and porous substrates. First, I would like to say a few words about our company. Fluicell is based in Sweden, and we offer products and services within life science field, drug discovery, and bioprinting. Our product portfolio includes bioprinter platform, Biopixel, which will be in the main focus of today's talk. Before introducing Biopixel, I would like to say a few words about bioprinting in general. So the core of this technique is similar to plastic 3D printing. However, the essential difference is that bioprinting operates by using a suspension of living cells instead of plastic filaments. So typically, the cells are housed in gel matrix or bioink, which is deposited using a layer-by-layer approach to build a tissue construct with the aim to imitate the behavior of native tissue. So to enable an optimal functionality of the printed tissue, a printing resolution allowing to control the cell-to-cell distances is necessary. So here at Fluicell, we address this challenge by utilizing microfluidic approach and generate high-precision tissues. This image shows the core of the Biopixel platform. This is a microfluidic print head which is inserted into a holder, and you can see a print tip right here. So using microfluidics for bioprinting allows to avoid usage of bioink, so the cells are housed in suitable culture media or buffer. And with this technique, it is possible to generate high-precision tissue constructs in 2D and 3D with a printing capability ranging from single-cell arrays to multi-component tissues. It is also possible to generate tissue models on solid and porous substrates, which I'll be talking more about today. Our Biopixel technology conveys high-resolution cell patterning by circulating cells within a confined volume close to a substrate surface, maintaining them in native media throughout. The substrate can be either solid or porous, or it can be a confluent layer of cells. Through patterning of a substrate with the appropriate proteins, control over the 2D and 3D patterning can be established. This particular example shows formation of a 3-layer construct where we started with the preceded confluent layer of cells, on top of which we print cell attachment agent, which is required for efficient cell adhesion. This was followed by patterning the second layer of cells, and by repeating this process, we deposit the third cell layer. The lower panel here shows example of a 3D pattern structure, which is basically a skin cancer model. Here, the base layer was composed of confluent layer of cells, which were A431 cells shown in green. The middle layer being Haycat cells, they are shown in red, these are keratinocytes, and the top layer being A431 skin cancer cells. With this microfluidic approach, it is possible to acquire a high control over cellular microenvironments of generated in vitro model systems. Such a control is a key to influencing cell behavior and producing physiologically relevant tissue models. And even though these models are representative of biological systems, they remain constrained by the bounds of the platform in which they have been formed. So releasing these bounds would open up a possibility for downstream analysis, as well as fitting them into other workflow processes. Freeing the tissue models from the confines of a culture dish can be accomplished through the employment of biocompatible porous membranes. These membranes are of particular interest as they enable for enhanced access of nutrients while also being suitable for regenerative medicine applications. So one good example here is the usage of polyethylene terephthalate or PET membranes, which have been successfully utilized in vivo and even phase 1 clinical study in age-related macular degeneration. The concept of membrane bioprinting can be generalized in 3 distinct steps. The first step is to bioprint cells to form biological tissue using suitable porous membrane. The source of the cells can be either from culture or obtained directly from a patient. After this step, the membrane can be incubated to allow for the tissue to develop and grow. In this second step, the microporous structure of the membrane enables nutrients access from both sides of the tissue. And after tissue maturation, the membrane could be excised for use in research or therapy. Here we show examples of 2D and 3D bioprinted skin cancer models on semi-permeable PET membrane. This is an example of a 2D model where 2 different cell types, Haycut keratinocytes, shown in blue, and skin cancer cells, SKML28, shown in red, were printed in a square shape. And this is an example of a 3D tissue construct, which was built using a bottom layer of cells, 3T3J2 fibroblasts, on top of which a layer consisting of 2 different cell types, Haycut, shown in blue, and SKML28, shown in red, were printed in a square shape. In this example, only the upper layer of cells was fluorescently labeled to enable easy identification. The images show the tissue composition right after printing and after 24 and 48 hours of incubation. And we can see that pattern cells form confluent tissue within a short period of time while preserving the pattern. To infer the suitability of the tissues for prolonged studies, we generated basic liver cancer model on porous PET membrane, consisted of HepG2 liver cancer cells and 3T3J2 fibroblasts. The functionality of a 2D liver cancer model, consisting of 2 units, each being similar to the one shown here, was probed by measuring albumin secretion over 23 days, indicating the suitability of this model for prolonged studies. On the panel on the right, you can see our recent demonstration where pancreatic beta cells from Min6 cell line were successfully printed on PET membrane. We believe that by employing the presented technological advantage to the formation of microtissues has great potential to broaden the applicability and utilization of the tissue models. Forming complex, multidimensional tissues on porous substrates should allow for more directed approaches to transition from the in vitro through in vivo and into the clinical field. To enable us to transition to in vivo applications, we are looking for partners to take this exciting journey. Here, I would like to introduce BioArray Advance, which is our recently announced partnership program for microtissue-based regenerative medicine research. We have initial focus at such areas as cardiac muscle repair, diabetes, and ocular regenerative medicine. We are actively seeking for partners to develop in vivo applications in this area, so if you have any interests or questions, please contact us. And with this, I would like to thank you for your attention and hope to hear from you soon.
Great. Thank you very much for that oversight into Fluicell, our sponsor. So with that, I'm delighted to close this session and thank all of the speakers and Fluicell for sponsoring. Please go and visit the poster session. It starts in 4 minutes, and then we'll see you back for the final session at 3:45. Thank you, everybody.